So in the previous videos, we have looked at how to implement analysis of variance to figure out if in one factor, two factors, or in combinations of several factors, there are levels which are causing statistically significantly higher level or lower mean values than other levels on the same factor, therefore, or the factor combination. So therefore, analysis of variance is very, very powerful if we are investigating the effects of different parameters, for example, on our analytical methods or in other chemical applications, also in manufacturing, synthesis, whatnot. However, analysis of variance can also be used further in quantification. And let us now look how analysis of variance concept can be used also when we are studying linear regression. So first, we need to, from our knowledge from linear regression from the first lecture, remind that we have different variances there. So basically what we have, I'm going to go to the next slide first. So basically what we have is a calibration graph where on the y-axis we can have a signal and on the x-axis we have a concentration. And here we have a mini example of a zoom in on one specific concentration level where on one concentration level I, we have performed measurements. The measurements are marked here with the black dots. There are 4 measurements visible here, and they all have their own specific signal values here on the y-axis. The blue line here indicates us the linear regression itself that has been fitted to the data. Not only this point, but all points that are there, we look only at one. And we can furthermore here add the mean estimate of the signal at this concentration level, and this is then this yi over here, which is then the mean across all the 4 individual measurement points. And from here we see that there are different effects or different variances. First of all, the 4 individual measurement points that we have are deviating somewhat from the mean estimate. And we can say and think when we make a comparison with also different factor levels in the one-way analysis of variance, that this is primarily caused by the experimental repeatability. So we have repeated the measurements. They are somewhat different from from the point estimate of the mean that we have gotten. And this concentration we can at the same time sort of see as one factor level. So our factor here is a concentration of the analyte, and specific concentration levels we can think of as factor levels. And in this setting, we have in our signal scale different variances. We have the total or sum variance, which means the variance across all points relative to the mean signal of the whole calibration graph. So not on specific calibration graph level, but across all of the calibration graph levels, all measurements that have been done. So this is basically like a horizontal line in our calibration graph, and how the different points at low concentration, they are lower, and high concentrations, they are higher. And therefore, this indicates how much in total the signal of this calibration graph measurements is varying. Then we also have part of this total variance that is explainable by the change in the concentration with the linear regression. And for this, we need to compare the signal values corresponding to the concentration levels on the calibration graph. So the predicted signal values corresponding to specific concentrations, and these predictions are made with our fitted linear regression. And when we compare how these are varying relative to the mean signal across the whole calibration graph, then this gives us how much of this signal is explained. So if the points are really completely randomly varying, the concentration does not have any impact on the signal, then the optimal line is also a horizontal line, and the explained variance is actually 0. However, if there is a perfect fit, then there is no experimental variance. Then the linear regression will explain everything of the total variance, and the explained variance and the sum variance will be the same. However, in practice, there are two sources of residual variance. There is the experimental source, and there is the lack of fit, meaning that the linear regression might not ideally describe the relationship between the signal and the concentration. And what is left over here, or this so-called epsilon term, which was in one-way analysis of variance, is the difference in the experimental values to the point estimates. And these equations here do not account for the replicates. So if there are replicate measurements, then this needs to be brought in as the different measurements on the concentration level I as well. And then, yes, the residual here, if we have replicate measurements, is the difference to the mean value. But if we have only one measurement, then it is the difference of the line to the specific measured signal value. Now, because the residual variance can be broken down further to model insufficiency and to experimental variance, in this case, we, however, always need to have replicate measurements. And in this case, the model insufficiency we can calculate as the difference in the mean value from replicate measurements, which we treat as an unbiased signal estimate for this concentration level. And here on the blue line corresponding to the same concentration level is then the predicted signal. The difference in this, showed with this brownish line here, is then what is the model insufficiency. However, the experimental variance is what is shown here more on the left-hand side. So what is shown here more on the left-hand side, how the individual measurement results are deviating from the mean. And in practice, both the model's insufficiency as well as the experimental variance are impacting how far off from the calibration line our measurement points are. Now that means that we basically have 5 types of variances, or one total or sum variance, one explained by the model, and then either a total residual variance or then this residual variance broken down to model insufficiency and experimental variation. And from this, we can ask 2 different questions. First question is what is corresponds to so-called goodness of fit test, where we are asking, is the explained variance statistically significantly larger than the residual variance? And in this case, of course, in calibration graph, if we are measuring, doing our measurements correctly, the measurements are done on a right wavelength or at the right m over z value, then the explained variance will be much larger than the residual variance. And therefore, the hypothesis that the explained variance is larger than residual variance is likely to be accepted in the hypothesis testing. The goodness of fit, therefore, in calibration graph case is not very often used, but it is very much used when we are using linear regression or other regression models in modeling other parameters for which we don't necessarily know if they are so clearly associated with the X values. For example, if we are studying retention time, linear regression with the polarity log P's of the molecules. Now, what is more useful in the quantification setting is the lack of fit. And this is the comparison now of the two residual variances. It is the model insufficiency and the experimental variance. So if the model is not able, if the model has a systematic error in comparison to experimental variance, then the SS or unfit should be much larger than the experimental variance shown here. And therefore, this is also the comparison that we are doing with the lack of fit test. So here the null hypothesis is that the model unfit is explainable by the experimental variance, while the alternative hypothesis is that the model residual variance arising from the lack of fit, so the model insufficiency, is larger than is explainable by the random variance in the replicate measurements alone. And therefore, lack of fit is very useful if we want to know if our model is actually, if linear model is actually sufficient for our, for fitting our calibration line. To go a little step further, we can also use analysis of variance type of thinking in choosing different models. But let us first clarify a little bit of statistical terminology. Namely, when we would have one measured, let's say, intensity or signal that we would like to associate or test modeling through different parameters, here X, Z, and maybe some residual, or sorry, some coefficient in additional, so the intercept type, or we would like to fit the X and X square, a model with an intercept, then even though in math class, we would usually say that the first one is a multilinear and the second one is then a squared equation, then from statistical point of view, these both are linear models. And this is because in statistical fitting, we are solving the equations not from the X and Z perspective or X squared perspective, but from the coefficients A, B, and C perspective. And because they are all in the power of one, therefore these are considered statistically linear models. Now, when we are fitting a model, for example, in calibration, we usually fit the linear model because we know this is how the chemistry and physics behind suggests. But sometimes it looks a lot like having a concentration squared, so a square component could benefit the fitting. There are two considerations here. One of them is if it is chemically or physically logical to add such a component, but the other one is also statistical. Is this really a component that is needed, or is this so-called overfitting our data? That means that it is fitting too well to the data that we provide during fitting and does not really generalize for the real conditions, so for the samples, for example. The more, generally speaking, the more simple the model is, so less parameters are included, the more robust these models are. And basically, the simplest model is considered where the output, let's say signal in calibration graph case, is just predicted to be a constant value, independent completely of the parameter concentration here. Now, when we start adding more coefficients, this has only the intercept. Maybe when we had a concentration term and the coefficient B here, then the model becomes more and more complicated. So where could we potentially want to compare models with with different number or different type of parameters? Well, one of them is if a calibration line should actually include also an intercept. Is intercept actually improving the model? Then we can also ask about the concentration squared term in the quantification case, or adding other terms when you're doing more complicated modeling. So, for example, if we have a signal intensity explained by concentration, and let's look at here on the equation side and more generally, where we have a large y, which you can think of as a concentration in terms, as signal in terms of quantification graph, and x as concentration in this case. Then we could have a model where the y is x is completely dependent on x. This would be when the concentration or mass would be directly out produced. For example, we can think of this as weighing. We put different weights for which we know on the balances, and then we take a recording, and this could be like a simple model. Then there are cases where the intercept is not needed, but the slope is needed. There are cases where the intercept might be significant, or we can have also the case, which is very often for us, where the slope and intercept are possible and maybe also important. So it should be thought if intercept makes sense as well, and compared to this above equation here without the intercept. And then we can also have the square terms with or without the intercept included. In these equations, there is a different number of coefficients that need to be fitted, and also the degrees of freedom are different. Also, this impacts how many data points even are statistically needed to fit such an equation. So the less degrees of freedom, the more measurements are needed as a minimum. Now, when we are, for example, investigating the possibility to do one-point calibration in terms of omitting the intercept, then we could, as one of the first questions, ask, is the intercept needed? So that means that we have two models, one with the slope term and the residual error, and the other one which also has the intercept term and also a residual term. However, even if the measurements are done on the same concentration levels and same signals are used, which should be the case here, then the residuals are not exactly the same, because on the lower equation, the part of this residuals might or might not be explained by the intercept term here. In both cases, we can calculate the variance of the residual variance not explained by these models here, by looking at the sum of squares across all the points that were used for measuring, and then taking into account the degrees of freedom. The degrees of freedom are more when we have only one coefficient, and by one it is less when we have two parameters that are fitted. Now, the question is, or the assumption is, that the more parameters we're adding, the more variance is being explained. So we can also, from the sum of squares, calculate how much the more complicated model, so the model with the intercept, is explaining variance more. So that becomes the difference in the sum of squares of the residuals for these two models, divided by the number of degrees of freedom that is different between these two models. So here it's just one, because one coefficient is added. And now we want to basically ask, is adding this intercept actually making the model significantly better? That is the variance that is additionally explained by adding intercept statistically significantly larger than the remaining variance, so the variance that remains when we are using the more complicated model? So does adding intercept substantially improve the fit of this linear line? And this becomes again a comparison of the variances, like it is in case of analysis of variance, but here we are comparing additionally explained variance with the remaining variance for different models. And last but not least, let's look at the case study for applying such analysis of variance for the calibration as well. And let's look at the very simplified data set where we have 5 concentration levels and we have on each one measurement with its peak areas. And we have here fitted 2 lines. The pink line is without intercept and the blue line is with allowing intercept. Now the question can be which one of them is the more correct one to use. Well, of course, we should first think about what is chemically making sense. Should there be intercept or not? But from the statistical perspective, what you have already looked at is to use an LM function, so fit the linear model, and you also have looked at the summary file, which gave you, among other things, I'm here zooming into the part where we have the coefficients, we have their values, which are point estimates, we have their standard errors, t-values, and there are also the probabilities. And what we didn't very specifically cover before, where do these probabilities come from? We only said that if our significance level is 0.05, then we clearly would see here that, yeah, the probability of getting such a point estimate as intercept is larger than the significance level, and therefore we say that we stay with the null hypothesis, saying that the intercept is actually insignificantly different from 0. However, we could also think of the same question as 2 different models. The first one with intercept, which we can again fit with the LM as we did before, and one without intercept, where the LM function is a little bit different by us omitting the intercept with writing -1. And now we can use actual analysis of variance to compare these 2 models, which would then compare the additionally explained variance by a more complicated model with the remaining or the residual variance of the more complicated model. And here, as the result, we would see also the analysis of variance table. We see what are the 2 models that are being compared. And we see both the residual degrees of freedom as well as the degrees of freedom of this comparison. And then the residual degrees of freedom here. And then what we also want to look at here is the probability, which comes from the F distribution. And here we clearly see that this, again, is higher than our significance level. And actually, what you might notice is it's also the same probability as what we had before in our LM function table. And actually, these probabilities are coming from such an analysis of variance for different coefficients in the linear model. So with this, we become also able to statistically significantly evaluate if a specific coefficient should or should not be used in our models.